All right, hello everyone. Thank you very much for Sourav Mukherjee, a PhD student from the SKB lab at the School of Biological Sciences at Monash University, for his time for Expertise Episode 6. So, Sourav, why don't you introduce yourself?
So hi, this is Sourav. I'm a third year PhD student at School of Biological Sciences in Monash. Trying to investigating on understanding how plants sense temperature using a model plant, Arabidopsis thaliana, and my research is going according to the plan what I actually thought initially when I joined the PhD, and it's good fun exploring different genetics and epigenetic across, and definitely working with plants is fun.
Interesting. So, Sourav has been my lab mate for 1 year now. So, here comes the most important question of all. Sourav, what do you think of me as a lab mate?
Oh man, come on. That's a very, well, let me think. Michael has yes, it's been 1 year actually.
It's been 1 year,
it
yeah.
Feel like it's time just flies.
Time flies, yeah.
Yes, as a lab member, you are extremely energetic. You bring positivity to the labs.
Okay.
Of course, not only in terms of science, also as a outside science in the lab or the office environment, you brings a lot of positivity, and you spend a lot of times in the lab, man. I should say that. So definitely, you are a very positive addition to our labs.
Thank you very much. So in conclusion, am I a good lab mate?
Nah. No, I just kidding.
Cut it. Oh, this is going to be a really good cut for the trailer later. Like, we can cut it later.
Yeah. Nah.
But
jokes apart, you are a very good addition to the lab.
Thank you very much.
Yeah. And thanks for being the part of the SKB lab.
Thank you for your opinion. Oh, okay. So, can you tell us more about your PhD project? You mentioned that you are understanding the genetics and epigenetics of plant response to temperature, right? So, can you elaborate more on?
Absolutely,
what?
Yeah. So, like if you ask me like what I'm doing in my PhD, the one-liner answer would be I want to understand how plants sense temperature. So the bigger goal of this project is basically you understand the genes and the regulatory networks, which basically involves in sensing or basically following the temperature cues in terms of different environmental conditions, so that you actually come up with modifying the plant in such a way so that you want the plant to be behaving that way what you want it to be. In the context of climate change and global warming, it's a very important thing, and as you know, in response to basically every year the Earth's temperature is rising, and the problem with that is basically plants. We have seen that increasing temperature is actually correlated with reduction in crop yield. So we really need to do something to make sure we are not getting reduction in terms of the yield of the cropland. If actually we need to increase the yield further, because the population of the world is also rising. So if we can't do things, it would be a huge problem in the coming years. And that's why the researchers across the globe started mostly understanding the temperature in the context of there is I should say one thing. There is another thing called heat stress. So this is not what I'm actually interested in investigating in my PhD. This is more about the growth of plant in ambient temperature differences. So this is different from heat stress. So this is basically where even 1 degree changes in temperature can lead to different phenotypic changes. And yes, of course, as you said, my project involves understanding basically investigating other novel genetics component of the temperature response pathway, and also going towards the mechanism side of things, where basically epigenetic science comes into the pictures. So yes, it's epigenetic gene regulations what I'm investigating currently, and hopefully by the end of my PhD I should have a mechanism to basically go forward further on that side of things.
So, would you mind sharing us the main findings from your project so far?
Yes. So, as I told you, I work with genetics. So there is a very popular terms in the context of genetics research. It's like a genetic suppressor screening, and we did that suppressor screening, and from there we basically picked up novel suppressors, which the idea of doing that experiment is to identify novel genes which are involved in sensing temperature. And so far we are actually in the process of identifying them. So we had a clue, like we had like a list of like, let's say 10 or 15 genes as of now, and we are basically chasing each one of them, and the experiment is currently underway. So I don't want to reveal the name of the genes as of now because it's
still secret. It's very secret.
It's a secret as of now, but hopefully I'm hoping in like few months we should be able to get it passed through, and then we can tell the world, look, this is the genes.
Once the paper is released, we are going to put the link in our YouTube as well. So viewers who are interested, they can come again in like a few months and see the paper there. But anyway, so later, what are you doing with these genes? Are you trying to knock them out, or?
Yes, currently I'm basically, yes, that's the thing. I'm basically currently knocking them out in the wild type plants to see whether they are actually indeed responsible for involving in temperature response pathway or not. The idea is if they do, we should see a phenotypic changes if you grow them in, let's say, 2 different temperature.
What kind of phenotypic changes would you expect?
Generally, as I told you, I work with Arabidopsis thaliana, which is a model plant. So there are like several phenotypes which generally the temperature, we know that induces due to changes in temperature, among which hypogotyle is one of them. So that is known then in the wild type plants. If you grow the same plant in, let's say, 2 different temperature, let's say 23 degree and 27 degree, 27 degree you should see the hypogotyle got elongated compared to 23 degree. So this is one of the phenotype. Another phenotype which we know that the flowering time, which is pretty popular across the crops and everything. So it is also known that temperature induces the flowering. So if you grow the same plant in 27 degree, you see that 27 degree the flowers are there.
So about all of these research you've done in Arabidopsis, would you say that it's applicable to other plants as well?
Absolutely, that's the whole purpose, because it has been shown that long ago that most of these genes are basically conserved, like their function is conserved across different crop species, and it has been also shown, like, let's say, it's not on top of my head right current now what is the exact gene, but researcher has shown like taking a genes which has a function in Arabidopsis and putting the genes in some other plants to basically rescue the same function, and it actually works. So do we know that the gene which we are actually having a function in Arabidopsis, that research can be translated to other crop plants, and that's the reason why we actually started working on Arabidopsis, because it's much more harder to work with crop plants because the genome is more complex, whereas Arabidopsis genome has, like, let's say, this has a huge advantage of like small genome, doing genetic manipulation quickly, easy to do, not that much of gene duplications compared to the other crop plants. So that's what makes Arabidopsis more popular among the plant research as a model plant.
Interesting. So apart from these, what kind of phenotypes would we expect in other plants? Like, let's say, like one of the main concerns about global warming is the decrease in crop yield, right? So in these crop plants, what kind of phenotype would we expect when the temperature increases, apart from crop yield, probably?
Yes, generally the 2 things people are more cares about at this point of a time, so I should say, is the flowering time, because you want the plants to flower early so that you can get the things quickly. Another thing is basically the growth. You can do the growth faster. Although actually, as I told you, in high temperature you see a faster growth as well as early flowering, but what you actually see is the reduction. At the end of the day, it's a reduction in the crop yield, because it's although it's not a stress condition, but still there are some level of stress. I don't know whether the stress is the right word, but there are some levels of response pathway turned up at that temperature, and that actually leads to this crop reduction at the end of the day. So it's a challenging work to basically fix that problem, and I think it's not that, I mean, even if we discover, like, say, one gene in Arabidopsis, it's not like if we just eliminate the genes from the real crop, we can actually solve the problem. It's much more complex than that. And in order to do that, I mean, the one thing we can do is basically try to fix it. We don't know what will actually happen because the real crops are much more complex to manipulate the genome. We have to start. We have to start, and we have to give a try to basically fix the problem. And that was the thing. That's the more motivation comes to these pictures to basically identify the entire regulatory pathway, so that you can know what are the entire things is doing, and then you can actually try to fix the problem rather than picking one gene, because you know about that gene but you don't know about the entire pathway. So even if you remove that gene, some other things comes into the picture and compensate for that.
So that's redundancy.
Redundancy is another factor. Yes.
Okay. You mentioned earlier that in response to higher temperature, plants potentially have a higher rate of growth, faster growth,
yes.
and also an earlier flowering time.
Yes.
Can you propose like a evolutionary advantage of this adaptation? Like, why would plants, when they are grown in higher temperature, exhibit these phenotypes?
Well, that's a tricky question, and actually it's actually out of my current research domain, because I don't think everything in the context of evolution to the extent of like evolutionary biologists. They probably would be more suitable for this kind of a questions. But yes, plants are very the plasticity of plants, because the idea we had was, okay, we human can basically move to other places and go into our comfort zone, but plants can't. So wherever you keep them, and whatever difficult or extreme stress condition you put on them, they always comes up with something to deal with that. So I'm pretty sure even if you try to grow them in much higher condition, they will grow, they will try to survive. But the problem is when they are trying to survive, it actually basically compromising on the other things, right? So the crop yield, one of them. Yeah. And that's the most important thing in the context of food security, because the world is like, now the population is rising, and then you have this temperature, global warming, climate change is a serious problem now. So we have to really come up with what we call it as climate smart crop, so that can deal with any kind of extreme conditions without compromising on their crop yield, so that actually the food security remains like food security remains like not a major problem in the upcoming years. And one of the way to deal with is basically try to understand how plant is actually doing that in the molecular level.
Makes sense. And about the food security and global warming.
Yeah.
This is a major concern, right? So, is there any way that is there any approach we can take to combat this issue of food security in the upcoming years? Well, overpopulation and also climate change. Probably, if you have any kind of insights or suggestions that.
I reckon this is questions where people have different opinions, and there are a lot of opinions coming from different people. But that's one of the thing I basically because yes, one side you have a like population increases, another side you also have to increase the food production on the crop to basically make sure everyone is getting the food. But here is the problem is, you are not only able to secure whatever actually you are getting before, because the food, instead of increasing the food, you actually have a reduction because of the yield. And also genetically modified crop is a like we try to basically whatever research we are doing is basically making genetically modified crop, but like not all the peoples are open about that, and there are some issues regarding genetically modified crops acceptance among the people. So I think we have to go basically talk to the people more to basically say that what we're actually doing is basically explain them in a more simpler way, not like so much about so-called scientific way, rather than basically, because I feel like most of the time the people because there are a lot of misinformation in the internet as well as among the people. So that also creates some level of problem in the context of accepting the genetically modified crop. So that's another thing. I think talk to people, try to convince them, try to tell them what we are actually doing, and if we can tell them in a more simpler way and what we are actually trying to do, I think this problem of like not accepting the genetically modified crop can be solved to some extent, if not totally.
All right. So we're also thinking about. Have you heard about C3 and C4 plants?
Yes, I do.
And these are basically different approaches of carbon assimilation in plants that live in different temperatures, right? So we expect that C4 plants tend to live more in higher temperature because they have a higher affinity PP carboxylase enzyme to compensate for the lower stomatal opening during the higher temperatures. And if this trend of global warming, increasing atmospheric temperature continues, would you say that in the future plants will somehow, the evolutionary trend will promote C4 plants instead of C3 plants? Like,
well, it can be, and again, I should say that this is not something of my area of interest or something which I am working on. I know, like, let's say our, like, in our department, Maria is like one of the faculty. She's working on the C3 C4 plants because I saw one of her presentation before. But what I know about, like, in terms of temperature and carbon dioxide is it has been shown in plants, in, like, let's say in tomato. There's a paper came in like a few months ago where they said like elevated temperature actually increases to the temperature sensitivity to the plants, because they saw like when they increase the CO2 levels, the plants are more sensitive to temperature rather like compared to when there was no CO, like less CO2. So definitely that gives us a clue. I think probably CO2 is maybe there is a possibility of a crosstalk between temperature sensitivity and CO2 in the context of temperature dependent growth in plants. And there are a few groups. What I know is basically doing research on like three combination of things. Let's say one is CO2 level, water stress, and the temperature sensitivity in plants, in starting from Arabidopsis to tomato, and probably many more. But to address your question, I think I'm probably not the best person to give you insight on that area of interest, because it's definitely not in my area of interest. Yeah.
probably we'll ask Maria for this.
I think that's a very good idea to do. Yes, I highly recommend that.
Alright, so now we come to our project. So our project basically plans to take a single-cell eukaryote called Pichia pastoris. It is a species of yeast.
Okay.
It is originally a heterotrophic yeast, but we are planning to turn this yeast into an autotrophic yeast by inserting genes responsible for the Calvin-Benson CBB cycle.
Okay.
And then the excess glucose we'll try to use to drive the production of an anti-mosquito compound called epictalotone.
Okay.
So when we use the carbon dioxide for photosynthesis, they create more glucose, and the glucose can be used to repel insects, basically. Because when there's higher temperature caused by carbon pollution and greenhouse gases, we will see a larger distribution of mosquitoes and other disease-bearing insects. So we think that if we can use this problem of carbon emission, air pollution, and greenhouse gas and turn it into a solution for another related problem, then this device, this engineered yeast, can kill two birds with one stone.
Exciting. That's an exciting proposal you have, I should say. Yeah. And
Will you say any challenges regarding this project?
So you are taking the, if I understand correctly, you are taking that strain of, I forgot the name, what you said, like
The Pichia pastures.
yes, and then you are basically trying to introduce. How do you actually introduce that to that system?
That's what we are going to ask you. So, because you've been involved in like genetic engineering and genetic manipulation of organisms, although we're talking about plants, is there anything you can propose to us of the way of incorporating these genes for the metabolism of serotonin into the yeast?
So they don't, if I understand correctly, that strain doesn't have that gene
No.
there, right?
Yeah.
Well, of course you can introduce that gene with a, like a, let's say a 35S promoter or another promoter which can actively expresses that gene to the system, but the major and also can integrate to that genome. But
How will be integrated to the genome?
because you can use like the it will probably it will not integrate to the genome, but you can definitely express that using a 35S promoter
Probably in the plasmid.
in the plasmid. But the major challenge in that process would be how do you, because that will probably not going to integrate to the genome. So even if you do what you want them to do, the question is how long, because definitely when you are trying to introduce a new gene to their system, they also have their own mechanism, like the own, like that strain also has try to basically not, there will be probably my understanding is there will be some kind of a resistance to address that questions. And if you actually, let's say if you have the genes, if you wanted to put it with a 35S promoter, what would be your control in this case then? How do you actually, what are you going to compare once you introduce the gene into the strain?
We wanted to cause the yeast to be able to photosynthesize and also to create this beta-lactone compound.
And how do you actually quantify photosynthesis or something?
Good question. We haven't thought about it because our plan was for that microorganism to be housed in a bioreactor,
Yeah.
and that it will create a beta-lactone that will diffuse away and can be extracted afterwards. And that's all our project is. We don't quantify the amount of photosynthesis, so we don't really. In simpler terms, we don't really care about how much the photosynthesis yields. We only care about how much beta-lactone it produces.
How do you actually quantify that compound?
The extracted compound.
Yeah.
But we can weight it and determine how.
No. How do you extract that? How do you actually purify that, or from the entire thing? Yeah.
Well, this compound is a small non-polar one, so it will just diffuse away from the yeast, from the cells, so it won't. We don't have to lyse
But
them.
what if let's say together with that it actually diffuses something else which we actually don't know? How do you? I'm more thinking on the direction of the purity of the compound, or because
The purification
of that compound, because it it maybe it can, like in a more like critical way, it can basically, whatever you are trying to do, if you introduce that gene, you can see whether that thing is working or not. But how do you actually make sure that it's only doing that thing and not doing something else which can be a problem in the future?
I think first the extracted mixture of beta-lactam and other diffused compounds,
Yep.
we'll probably check what's inside first. So, like for example, let's say it diffuses some kind of other enzymes probably, and then what we're going to do afterwards, probably try to purify it by passing it through, let's say, some kind of affinity column chromatography. So it will actually attract other proteins. That it will basically remove all proteins, and then there will be this mixture of beta-lactam and other solutes probably, and then we can pass it through some other filters or some other selection process to actually, in the end, produce this pure beta-lactam that can be distributed to repel insects.
Are you planning to do like some kind of a negative control to use in this experiment?
No, because it's more of an application-based research, not a scientific-based research. If you get what I mean. Like,
Yes, I mean, but I think probably at the beginning to test your idea, I think it's a good idea to test it with a proper control.
Probably a proper control will be, probably the pure negative control will be an unedited wild type strain, and then probably another edited strain that only bear the photosynthetic genes but not the beta-lactam synthesizing enzymes.
I mean, you can definitely quantify, let's say, for example, one step before talking about the photosynthesis pathway, you can quantify one of the protein, or let's say the total protein from there, to see whether you see actually at the first place an increase in a protein level in one of the, let's say, one of the enzyme which is involved in one of the byproducts of the photosynthesis pathway. You can basically quantify them using, let's say, mass spec.
Yeah.
So if you do that, then you actually know that it actually increases that product yield of that photosynthesis pathway. Then you already know that it's actually targeting the photosynthesis pathway and it's actually seeing an increase in there. And then you can, that's an added line of evidence for you to basically make sure the thing which you are actually wanted to do is actually going with the plan.
Fortunately, all these are only like this competition only is based on design, so we don't actually have to.
Oh, you don't have to.
We don't have to actually do that. This is just a design competition.
Okay.
That's the reason why we are able to partake in this competition, because our team is composed of like 8 members and 2 supervisors or advisors from 4 countries. From 4 countries.
That's really exciting. Yeah.
There are some people in Indonesia. We are here in Australia, and then in the US, and also in Canada as well. So it is a huge international project. Fortunately, it's all online. If it's offline research lab based thing, we won't be able to do it. Honestly, it's crazy. But yeah.
But I should say, like, the idea is really cool, and it's like it's something I think you guys should explore also if you get a chance, and because this is a really nice idea, and I'm very glad like you guys have come up with this idea. This sounds so innovative, and basically it has, I can see a potential if you actually, but the only thing is, you have to explore to basically get to the stage whether you can actually implement
create it.
But given that the idea is always about, like, all the times we talk about ideas which basically makes your things happening. So if you do not have an idea, you can't start anything. So you have an idea, I think now is the time to basically test this idea, and if, who knows, if things happen like the way you are thinking, it can solve like a huge problem of the human.
Yeah, makes sense. So is there any other ideas for our project, like, for example, like extrapolation of the idea that you might offer?
Not I think the project idea is very strong according to me. I think this is like a really strong idea. I think the thing which I think you have to maybe think a bit more in, like, critically in some of these steps, and basically try to think about other possibilities. Not only thinking about, okay, this is the problem, this is what I have to do, and this can solve the problem. You also, I think, have to think about more on the side of, okay, if this doesn't work, what next? How do we troubleshoot each step? Or what can go wrong in another way that can lead to a problem. So this is like a more like a entire thing, not only thinking about the positive result, because in science there is nothing called positive result. Everything is a result, and I think you probably, if you think on that side, you might be able to find some area where you can actually do propose some more experiments, such as let's say the mass spec or something like that, to basically confirm after each step, to basically making sure you're on the right track, because that's very important in science, and it's actually making a lot more sense when when things doesn't work.
Interesting. So I think that's all from us. But to close this discussion session, we're going to put this recording on YouTube, and also
That's cool.
put some snippets of the recording in Instagram as our trailer. So our audience is mostly university and high school students in Indonesia. Do you have anything to tell them?
Yes, of course. I mean, when I was a student, like in high school, I always wanted to pursue like science and also application-based science, like basically whatever you guys are planning to do. Although I was not that smart to come up with this kind of idea when I was at your stage, so well done for that. But yes, it's something like if you really wanted to pursue a career on science, I think it's basically all about motivations to some extent. Particularly, I'm talking more of a PhD or a research side of things, because when I came, I was not having technically much expertise on the scientific side of things, but what I had was to basically a motivation to work on science and basically to work for something which can help solving some problems of humans and everyone's basically in the world. And other than that, yes, patience is one another thing. There will be challenging times in every step. I was like, let's say there are like failure is always will be there. I think if you like, I see this way, because if you see me succeeding like 10 times, definitely I must have gone through failures 100 times before. And that's people generally do not talk about, because people only talk about success a lot, so they actually stop forgetting to talk about failures. So failure is the important, like basically it will come anyway. Everyone has to face the failure, but the most important thing is basically to look at it as a lesson or as an experience, because technically speaking, I learned most of my stuff from my failed experiment, and that is the thing which basically helps me to succeeding when I actually get an experiment done. So don't stress too much if something goes wrong. Your grade, you are a bit disappointed. I understand everyone has an expectation on themselves, so sometimes things doesn't work, but it's just a part of the process. So don't stress too much on yourself. Just enjoy the science or whatever you are doing. Not necessarily has to be on science or anything you do. Just enjoy it, and yeah, just enjoy the process. I think that's the most important thing to do, and you should be fine.
Thank you for the wise words.